Okay, so without further ado, welcome again, everyone, to the start of the B-cell session and day 2 of our virtual conference. So it's my great pleasure to start this off with a description of antibody responses in SARS-CoV-2 infection. My name is Nina Luning Prak. I'm the chair of the AIRR Community, and I'm also a professor of pathology and lab medicine at the University of Pennsylvania. We started in this pandemic, like many others, analyzing patients with severe disease who came into our hospital. And we were very interested in trying to understand initially what kind of B-cell responses do these patients with severe disease mount? What kind of immune responses do they make? Do patients with severe disease exhibit any sort of typical characteristics in their B-cell responses or antibody repertoires? And what exactly is the frequency of B cells that produce protective antibodies in these patients? Now, we don't have definitive answers for any of these questions, but we do have a beginning point, I think, for several of them, not only through our work, but through the work of others. So the 2 early studies that came from our institution are shown here, and I'm not going to go through them in detail because there's way too much information here, but I just wanted to show these papers to you for reference. So there is an analysis of deep immune profiling of many COVID-19 patients using multi-omics platforms that reveals distinct immunotypes. That's one paper here, Matthew et al. And then the paper on the left here is the one that my lab was part of that I'm going to talk about today, where we are looking at immune repertoires in patients with severe COVID-19. And we began processing samples from these patients in late March, early April of 2020, and the data were collected and analyzed within 7 weeks. There were a lot of sleepless nights, needless to say. So the overview of this study is shown here. So patients came into the hospital of the University of Pennsylvania with COVID-19. We had an amazing faculty member within the intensive care unit, Noala Mayer, who was just incredible in terms of getting the clinical data together, getting all the protocols up and running, and providing informed consent, which was, it's quite challenging with these families going through this very difficult time to be doing all of this with us in the research arena as well. And we really owe all these patients and their families a large debt of gratitude. So we had 54 patients in the study, 12 of whom were healthy donors, and the remainder were patients with COVID who either had moderate or severe disease or who had recovered. These are not the same individuals in these different groups. The main distinction between severe and moderate was based on the respiratory degree of compromise and the need for invasive respiratory measures to support the patient. So all the severe patients, for example, tended to be ICU patients. Now, we obtained several samples from these patients. The one that we're focusing on here today is anticoagulated blood. Was processed fresh, usually the same day, and that was very useful because then we could do flow cytometry in lots of different subsets. So the Betts lab did the flow cytometry work, and this was on whole blood, including granulocytes. The Hensley lab did the serology assays, and my lab did the immune repertoire profiling on a subset of the patients. So we looked at 13 out of these 54 patients. And in terms of the repertoire profiling, what we did, whoops, how did that happen? Let me just go back, sorry. We took bulk DNA from cells that were not sorted, and we amplified heavy chain gene rearrangements with primers in framework 1 and JH, which of course recovers the CDR3. We used 2 by 300 base paired end kit, and the Illumina platform ran everything on a MySeq, as we've described many times before. So before I go into the repertoire data, though, I just want to give you a brief look at some of the immunophenotyping data that were obtained. So this was a 26-color panel. There were several different cell populations that were differentially expressed and seemed to correlate with disease severity. And so those are shown in the text here on the left. So there's certain populations that were decreased in frequency and others that were increased, notably neutrophils, eosinophils, certain markers of T cell activation, some cytotoxic CD8 T cells, and notably, of course, plasmablasts. And since we're focusing here on B cells today, I thought I would focus really on that, on the B cell population. If you look at the flow cytometry data here, you can see This is an example of a healthy donor versus a patient with severe COVID, gated on B cells, looking at the expression of CD38 versus CD27. In a healthy donor, you have a plasmablast frequency here of about 1% of the B cell gates, and in the severe COVID patients, it's over 40% in this individual patient, with also a large expansion of memory B cells. This is quantified here on the plot on the right, showing again that the plasmablast frequency in these individuals with severe, but also in some of the moderate patients, is quite expanded. Now, of note, a lot of the non-plasmablast B cell populations also exhibited abnormalities, including reduction in the level of CD21. So low levels of CD21 are sometimes associated with atypical memory B cell populations, which is of interest here because perhaps some of these atypical memory cells may be a manifestation of an atypical and short-lived immune response that may be extrafollicular. And in lines with that, several other groups, including notably Inaki-Sans from Emory, as well as Shiv Pillai from Harvard, have described an extrafollicular type of immune response that resembles SLE actually, and has expansion of certain double negative populations such as CD27 negative, IgD negative. And some of these double negative populations, notably DN2 cells, are actually poised to become plasmablasts in lupus and are associated with lupus disease flares. So it's interesting to speculate that perhaps some of these B cell abnormalities are similar to an extrafollicular type of response. So more on that in a moment. But now let's turn a little bit to the repertoire data. So for starters with the repertoire data, this is the AIRR community, right? So the first thing we really want to emphasize is that our data are publicly available. And so our data, this is a quote from our paper, the immunoglobulin heavy chain sequencing data are available and were deposited in an AIRR compliant manner on SRA under that accession number. We have also shared these data in the AIRR data commons via the iReceptor website. You can access our data, and that's shown right here. You can see there's a little announcement about it. We were already the 9th COVID-19 study to be entered into that database. Okay, so what do the patients look like for the IgH sequencing studies that were done? So this is a quick look at these patients. I think it's important to emphasize that these patients are quite heterogeneous with respect to their phenotypes, their clinical and demographic data. So what's shown here, we have 3 healthy donors, and then I'm just drilling down on the subjects with COVID. So there are 3 individuals with moderate disease, and then another 7 individuals with severe disease. You can see we tried to look at days post-symptom start. It's very tricky in these studies to time when disease is actually starting. And so there is variation in that. And then if you look at the age ranges of the patients, you can see also that most of them are either middle-aged or elderly. Then the oxygen therapy, which again is the main classifier for whether a patient is considered moderate but still hospitalized versus severe, is really the need for, in the case of the severe patients, more aggressive respiratory support. So you can see that many of the severe patients had acute respiratory distress syndrome. And you can also see now in this next column, vascular and metabolic, that refers to the presence of 1 or more risk factors that have been associated with severe disease, to include obesity, cardiovascular disease, hypertension, diabetes, mellitus, and hyperlipidemia. Also, several of these patients had other underlying illnesses that are significant. So for example, this patient had autoimmune disease, and this patient had B-cell lymphoma, but had B cells present, which is why when we sequenced them, we did obtain clones, but nevertheless, it's not a normal repertoire. So many of these patients may have some degree of immune compromise going into infection with COVID. Okay, so what did the immune repertoire data actually look like? So here's an illustration of that. So in terms of the data themselves, what we do is we have the disease categories here, the B cells, the plasmablast fractions. But remember, we're looking at global B cells here. We're not sorting on anything. We have the replicate libraries. So that's the number of independently amplified and sequenced libraries per subject, the input DNA, the copies, unique sequences, and total clonotypes. So the first thing that you can see from this table is just that there tend to be far more clones from the healthy donors than from the COVID patients. Part of the reason for this is that COVID patients can be somewhat lymphopenic. And so some of this may be due to limitations in sampling. However, a large part of it is also that the COVID patients tend to have more oligoclonal repertoires that are dominated by very large expanded clones. And to illustrate that, one of the easiest ways to visualize large expanded clones is to look at the top copy clones and ask what fraction of those most abundant clones occupy the overall repertoire. So to do that, I'm just showing you an illustration of this with two patients. One is a healthy donor and one is a patient with moderate COVID. And in these plots, we're looking at the top 20 ranked clones as a fraction of their copies in the sequencing library and asking what fraction of total copies does each clone occupy. So here the top ranked clone is 0.1%. In contrast, in the moderate patient, the top clone is 7.5%. Now in this circle, you'll see here, this is the sum of the top 20 clones and their contribution to the overall sequencing library. So here, the top 20 clones, if you sum their copy number percentages together, they come up with 1.6% of total copies are in this top 20 clone pile. So that's called the D20 measure. And you can see for the healthy person, that's 1.6%, so pretty low. And then if you look in the patient with moderate COVID, it's almost a quarter of all of the sequences are in just these top copy clones. If you plot the D20 index, each dot here is a subject, and you can see, of course, healthy donors tend to have low D20 indices, whereas patients with moderate and severe COVID have much higher levels of prominent clones. And then if you correlate the D20 percentage with the plasmablast percentage, now admittedly it's a very small number of patients, but the correlation is quite good. So it suggests that a lot of these high copy clones reside in the plasmablast pool. And in some cases, numerically, the top copy clone is so large, it can be 40% of total copies. It can actually exceed even the plasmablast fraction. So the top copy clones may even extend beyond the plasmablast fraction. So the next thing we looked at was just what these clones look like in terms of their mutation status. So somatic mutation, of course, gives you some insight into how an antibody repertoire or B cell response is occurring in response to an antigen. And with higher levels of somatic hypermutation, that you might tend to associate that more with a T cell dependent germinal center reaction type of response. And I know that one of our speakers is going to be looking at lineages, and and so Ken is going to tell us all about lineage tree analysis. Though the requirement for lineage trees is to have somatic mutations that you can study. And so looking at just the overall levels of somatic mutation is sort of one of the first things you do to study selection, is to get a sense of what that distribution looks like. So here we're just looking at these top copy clones because we think that those are the most interesting ones in terms of being associated with these potentially large plasmablast expansions. So if you look at the somatic hypermutation profile, here an identity of 1 means that the sequence is identical to the nearest corresponding germline V gene. So that would be an unmutated sequence. And then as you go from right to left, you get to higher levels of mutation. So in the healthy donor, the top copy clones, a lot of them tend to be unmutated. And that's also true, though, and kind of surprising to us initially, that we also saw that in the top copy clones in the COVID patients. So yes, they do have some expanded clones that have higher levels of mutation, but they also have a fair number of clones that are unmutated. And if you stratify the data by isotype, so these were not our data, these were data from Scott Boyd's lab that we analyzed by pulling them off of the iReceptor website. So this is an illustration of how data sharing can really help give us some insights, right? So we took the data from Scott off the website, and he shared these prior to even publishing this paper, right? So this is a great example of how how investigators in this era are really sharing their data publicly with the whole global community. So we took the data, ran them through our pipeline, and just asked, what level of mutation do you see? We counted each clone only once. And if you look at IgD clones, you can see that a lot of them are unmutated, have very low levels of mutation, whereas IgA clones have higher levels of mutation. And that's interesting because IgA clones also seem to be quite prominent in the early expansion phase of SARS-CoV-2 infection. So you can see by secreted antibody that IgAs are often quite abundant early in the course of disease. So, but they also have these unmutated clones in here. So even though these cells are class switched, you can see lack of mutation in some of the clones here as well. And these also were sequences from patients with COVID during acute infection. So large clones display a heterogeneous pattern of somatic hypermutation with a decent fraction of them being unmutated. Next, we looked at the CDR3 usage of these clones, and one of the striking findings that we had here was that the CDR3 sequences of expanded clones of the largest ones tended to be much longer. So this is just showing you the CDR3 length in nucleotides of the severe patients and the moderate patients compared to the healthy controls. And you can see that there's a whole tail of clones in the severe patients that tend to have longer CDR3 sequences. So longer CDR3 sequences are interesting to us because they can sometimes be associated with autoimmunity and polyreactivity. And so we wonder whether some of these large expanded clones in the COVID patients may in fact be polyreactive. We also looked for shared features in the CDR3s, and this is still a very preliminary analysis, and some of our other speakers are going to give much more interesting ways of looking at the data, so I'm not going to belabor this. But if you just do a very simple thing and make a logo plot and look at the CDR3 strings, they're not massive things that pop out of this initially. But again, there's much more coming, so we'll hold off on more detail of that. So in terms of sequence convergence, in our initial analysis, we have very limited evidence for convergence, but others have shown some evidence of convergence. So, so in terms of just looking at a low number of patients, it's tricky to do this, and there's a huge risk of false discovery. So if you look for things like whether certain V genes are overrepresented in patients versus controls, sure, you can find some, but the fold change is often modest and variable from one patient to the next. You can look at V gene family usage and stratify that by clone rank. And when you do that, what's interesting is that the severe patients tend to use more VH3 family members. You can also try to look at public clones. And so you can ask things like, are the clones shared with other known clones that are in public databases or shared with each other? So when you do that, you can see that there are public clones, and this is just identified. We're just calling clones here sequences with identical CDR3 strings. So they may not, in fact, even be identical clones. You can see most of them are shorter CDR3 strings. And that's, again, kind of what you would expect by chance also. So we don't have super good evidence in our data set of convergence. However, others have looked in their data. For example, Boyd has looked for clusters of clones that have certain similarities and has seen some evidence for convergence. And of course, there's a whole other massive effort underway that some of our other speakers are going to highlight for us today, looking at antigen-specific clones. And there is very significant evidence for convergence. So for example, in a study from a group in Beijing, where they isolated clones that bound to the receptor binding domain, there were very restricted V genes that were used in some of these clones that had high levels of neutralization titers, et cetera. So for example, they had certain V genes, 353, 366, and only 3 different kappas that were shown in just a handful of clones that they described. In another experiment, and perhaps some of our speakers will comment on this, there was a single individual who had a very dominant set of rearrangements that were characterized by single V heavy light chain combination. So sequence convergence is really a reflection of kind of the data set that you look at, as well as how you try to look for it. So in terms of initial impressions, I think what we have here are hospitalized patients with severe infection, and what we know is that they have significant expansions of plasmablasts during acute illness, as well as expansions of atypical memory cells. The plasmablast expansions are accompanied by large clonal expansions of antibody sequences that numerically can easily account for the degree of plasmablast expansion and are in fact correlated with the degree of plasmablast expansion, suggesting that these large clones reside in part in this plasmablast pool. The expanded clones have elongated CDR3 sequences and variable levels of somatic hypermutation. And these things kind of make us wonder whether some of these clones are being pulled out of pre-existing clones that may be multi-reactive and are just kind of available to be stimulated, potentially as a consequence of bystander activation in the setting of severe inflammation. Also, in terms of the proportion of cells that may be part of sort of the useful immune response, this may be actually a surprisingly low fraction. So, for example, the fraction of cells that target the receptor binding domain of the spike protein are at quite a low frequency, both in convalescent donors as well as in individuals with severe disease during the acute phase of illness. So if you look by flow cytometry, for example, and you use a probe for receptor binding domain, you take two different colors here, you sort on the diagonal cells, you can get, you know, a couple dots in a healthy donor, but so that's maybe 0.01%. And then in convalescence, you may get a 10-fold increase, but you're still only at about 0.1%. Now, some of this you could say, well, that's just because it's a small target, it's a small part of a protein. And you can do this same kind of sort with nucleocapsid or whole spike and get more events, but you still don't get a ton of events like you would in flu, for instance. So it makes me wonder whether part of the reason why when you look at the overall repertoire, you don't see the obvious patterns that you might with other diseases. Part of that may be because the fraction of responding clones, especially in people with severe disease, may not be as high in terms of being specific for antigen. So how do we put all of this together? And so I just want to provide kind of a general vignette. We haven't shown direct evidence for all of these things, so this is really just speculation at this point. But I think we can potentially think about these large clones as being a product not necessarily of a productive response, but potentially a maladaptive one, or a kind of all hands on deck response that's not necessarily T-cell dependent, right? So here I'm providing for you a possible way of thinking about this, that maybe individuals who have mild COVID have, you know, minimal tissue injury and have the luxury of potentially time to mount a protective T-cell dependent response that involves germinal centers, that provides adequate T follicular helper cells, and provides some degree of coordination between the T-cell and the B-cell response. And also in an individual with mild disease, perhaps we have higher clonal diversity, so that if antigen binding cells are relatively infrequent in the repertoire, then an individual with higher clonal diversity to begin with may have a better shot at having the right clones in their repertoire that can be expanded. Also in mild disease, you may have more affinity maturation, so higher levels of somatic hypermutation as a consequence of better BT collaboration. And perhaps you even have more on-target immune responses, although this is really very hard to say at this point. Serologically, people with severe disease actually mount very high levels of antibody responses, but how those antibody responses correlate to the cellular responses and the B-cell subset composition is not at all clear at this point yet. And then, of course, the other really interesting question is, how does this relate to memory? So if you look at severe disease in contrast to mild disease, what you see is potentially more of a pattern of high viremia, more tissue injury, potentially as a consequence of that viremia, but also perhaps underlying factors in the individual that promote further inflammation. Perhaps in severe disease, you have more of an extrafollicular immune response, and there are two studies that I've mentioned already. One from the group at Emory, commenting on how critically ill COVID-2 patients display lupus-like hallmarks of extrafollicular B-cell activation, including some of these plasmablast expansions and abnormal memory B-cell subsets. And perhaps some of these abnormal memory B-cell subsets don't fully progress to a full-blown proper memory response that's long-lived. And then, of course, the work from Shiv Pillai's group that was already mentioned at yesterday's meeting, where there is a loss of T follicular helper cells in germinal centers. And perhaps some of these other cell types, so also mentioned yesterday, could these cytotoxic T follicular helper cells be contributing towards a shift into a more maladaptive immune response that's oligoclonal, that has less somatic hypermutation, and has more instances of off-target immune responses. So with that, I think I'll stop and see if there are any questions. So let me just end the show here, stop sharing, and see if we can see the questions. All right, so give me a second here to work through. Okay, so we have some questions coming in from the Q&A. Okay, so the first question from Bjorn Peters is, Nina, how does the increase in plasmablast and atypical memory cells in acute severe COVID samples compare with other acute viral infections? That is a great question, Bjorn. It's interesting. So the plasmablast fractions are very high. You can also see very high plasmablast infections in dengue. I think there may be some descriptions of it in Ebola, some very severe, basically severe infections. You can also, however, see really high levels of plasmablasts in autoimmunity. So when I first saw some of these flow plots coming out, it just looked to me just like a lupus patient who was having a really severe flare. And so we have seen similar levels of plasmablasts in lupus patients. They can be over half the B cell gate sometimes, so really quite massive expansions. So and plasmablast expansions are not unique in any way to COVID in terms of, I mean, you can get plasmablast expansions with lots of stimuli that, you know, that aggravate the immune system, but the degree of plasmablast expansion is quite high. Okay, so then a second question, also from Bjorn, same question applies to observation of large numbers of unmutated clones. So what are the unique features of COVID and what are features of studying the acute samples? Okay, so that's a little bit trickier. So in our, and I'm gonna actually defer a little bit to some of the other speakers who have been looking at clones in other infections. So maybe we can hold off on that question for further discussion once we've gone through the whole panel, because I think that's a really cool question. So now let's see, we've got, did you look at plasmablasts? So this is from Yogesh Singh, I hope, from Tübingen. Yogesh, I hope I'm saying your name correctly. Did you look at plasmablasts in mild patients? We have looked at a few. I haven't shown you any of those data. They tend to have lower plasmablast frequencies than severe patients. That is what we see. Okay, from Lindsay Cowell, I noticed a lot of patient variability in the percentage of plasmablasts for severe patients. This is definitely, yes, that's true, Lindsay. We do get quite a bit of variation. And could this be due to differences in the onset of disease? Certainly a possibility. We did try to look for that, but there is so much variation that it is quite tricky to nail that down. And even though we've recorded the number of days since the start of symptoms is not necessarily a reliable indicator of the start of disease. So I don't have a really convincing answer for you for that. I think what we will need for that are sort of larger studies with larger cohort sizes. Okay, so an anonymous attendee, were the Illumina sequences validated with UMIs to exclude sequencing bias? No, they were not validated with UMIs. However, the way that we looked at the sequencing bias was to run replicate libraries. So in terms of these clones, these large clones, we can not only show that they are large in individual sequencing libraries where you would be concerned potentially with a jackpot, you can show also that the clones have a high degree of reproducibility across independently generated sequencing libraries. So the clones are large in multiple independent metrics of size. Okay, so now we've got Lindsay. Okay, so Lindsay comments that there may be a low somatic hypermutation in mild patients as well. Please reconcile this with some of the other observations. I think that's really fascinating. I have not seen those data, Lindsay, so maybe we can think more about that together. Okay, Nathaniel Spisak, I'm sorry, Nathaniel, if I'm saying your name wrong. Can you expand on how atypical are the memory B cells constituting the short-lived plasmablast response in terms of which characteristics they have that stand out? So we have seen that one of the most prominent features that we have seen is low levels of CD21. And low levels of CD21 are associated with some of the atypical memory B cell populations that you can see in chronic viral infections, interestingly. You can see that, for example, in malaria and HIV. But what's really interesting to me is that the CD21, the low level of CD21 is not unique to a single subset. It actually spans across multiple B cell subsets, which makes me wonder whether what we're seeing is something that's more global with respect to CD21 that we either don't understand, or that the subsets themselves are a little bit more fluid than we appreciate at this point. The other thing I will say is that we do see expansion of certain kinds of subsets, such as double negative cells, and these double negative cells are also expanded in lupus flares. Double negative cells are really interesting, and Inaki-san's group has done very nice work in recent years showing that a subset of double negative cells called DN2s is associated with, it's both transcriptionally and epigenetically poised to become plasmablasts. So it's really quite tempting to speculate that something similar may be happening in acute COVID. All right, so I think with that, although we are running a little bit early, what I want to try to do, we have lots of nice questions here, and I thank the audience very much for their engagement. I'd like to reserve some of the remaining questions for the panel discussion, if that's okay. Are there any other questions that people would like to ask live? No? So people like to do the chat with the questions. So this is an interesting experiment for us, and I apologize to all of you for subjecting you to being a bit of a guinea pig with this session in terms of how we work out the questions. But I think it's working reasonably well. So what I'd like to try to do next, I think, is introduce our next speaker, and move along with our program today, and then save some of the remaining questions for the panel discussion, if that's okay.